Okay, the term basic means it's on, it's off. So you look at an action potential that is basic, even though this stimulus is on, so this is on, and it's not off till here. What's happening with the neuron? It responds, and then it doesn't do anything until the stimulus turns off. So it's on for a little bit, so a phase, it's on, and then it's not responding at all. Some smooth muscles act like that. Some neuron receptors act like that. So phasic, the smooth muscles are going to contract in bursts. They'll contract when the stimulus is on, they'll contract when the stimulus is off, they'll contract again and turn off again. So there'll be bursts of smooth muscle contraction. As the action potentials are being generated in the smooth muscle surface, then calcium channels will open. Calcium comes in, binds to calmodulin, activates that complex, activates the protein kinase, and then you get phosphorylation of myosin head, cross bridges form. Tonic smooth muscle contraction means the stimulus is on. You turn on the stimulus, and the muscle continues to contract until the stimulus is turned off. It may not stay at this high level of contraction, although a smooth muscle can do that. It probably will taper off a little bit. So with a little bit of decrease in force until the stimulus is turned off. So you lose a little bit of force, but you keep most of that force applied in a tonic smooth muscle contraction. The calcium stays in the cytoplasm for a long period of time. You also have this myosin head attached to actin for a long period of time. You don't detach like you do in skeletal muscle. The ATP is there, but it doesn't, you have to have it transferred by the protein kinase. So in smooth muscle, it's efficient at holding on to that force of contraction without additional consumption of ATP. So it's a very efficient cell. Not only does it contract from all angles and apply force from all angles, but it can maintain that contraction for a long period of time. If you choose to have that tonic smooth muscle type of cell. If you want it to contract and relax, then you have a phasic smooth muscle cell. We have pacemaker cells, we have that are shown here. We have these cells that are producing what are called slow wave potentials. There's still smooth muscle cells, but one is setting the pace of the smooth muscle contraction in the GI tract. That's where these pacemaker cells are located. The pacemaker cell itself is in the interstitial area, and they're named for the Spanish guy Santiago Ramon y Cajal. Probably, if you're in lab, you heard his name. If you saw the video about the neuron contraction, I think that was the one that was broken. So I think one lab didn't get to see it because they jammed it in the VCR, and then the whole video got damaged. But anyways, Santiago Ramon y Cajal was a Spanish anatomist. He was the one that discovered or he proposed the space of the synaptic cleft. Previous to Santiago Ramon y Cajal, because I love that name, so I don't mind saying it over and over again. Previous to him, they thought that the neurons were actually connected to each other. And he said, no, there's a space there. And I do not believe he had an electron microscope, because he died about 7 years after the electron microscope was invented. So kind of doubt he had access to it. Can't find any references that say he had an electron microscope. But he's looking at things through a light microscope. That's pretty good intuitive thinking. It took a German guy to come along and actually figure out what the communication between neurons was. So, a name that I don't like to say, but anyway, it's a German, Otto Lurie. He discovered neurotransmitters, and then he had the way that neurons could communicate. Okay, coming back from that tangent, Santiago Ramon y Cajal also proposed pacemaker cells in the GI tract, so that the neurons from the autonomic nervous system did not set the pace for GI motility. That actually the GI tract set its own pace for smooth muscle contraction. He said, yes, the nerve probably influenced it, but there are pacemaker cells within the GI tract that set the rate of muscle contraction. So they got named after him. After they identified the cells that were pacemakers, then they named it the cells, the interstitial cells of Cajal. So what they do, they like the heart pacemakers, they have no real resting membrane potential. They fire, and then they repolarize, and as soon as they repolarize, they start to fire again. So they will have what's called a pacemaker potential, where they will slowly depolarize when they hit threshold, then there's a rapid depolarization, and then a fairly rapid repolarization. So they're continually firing. Other smooth muscles are responding to that. The slow wave potential is there's muscle tone increasing, but no actual action potentials being generated. So the membrane potential goes up, and there's very little background increase in smooth muscle tone. And as soon as you hit threshold, then these smooth muscle cells will fire a series of action potentials, and they will contract. And then you go back sub-threshold, and the resting membrane potential will oscillate. We'll apply this more next semester when we're studying the GI tract. So right now, you just need to be able to tell the difference between a pacemaker potential and a slow wave potential. So waves that are elongated, this is a slow change in membrane potential, slow wave potential. If it reaches threshold, you get action potentials sent. With pacemaker cells, they reach threshold, fire an action potential, repolarize, back to resting, don't stay there, just start to automatically depolarize again, hit threshold, another action potential is sent. So every time you get a change in membrane potential, it will reach threshold. In a pacemaker cell, it will fire an action potential. Here you have all these depolarizing potentials, but not everyone will reach threshold. Only the ones that do will actually fire action potentials. So multi-unit. These are individual smooth muscle cells that are contracting what did I say? Single unit. Okay, I told you wrong thing then. Sorry about that. Okay, wait a minute. No, I was right. Okay. I was like, what my brain is doing to me? Okay, so multi-unit, each individual cell is contracting independently of its neighbor. So it's responding to the neurotransmitter. The neurotransmitter must bind to a receptor on each cell for the individual cells to contract. So in small airways, large blood vessels, you might have the location of these single unit cells. So you want to control minute changes in the diameter of a blood vessel, the diameter of an airway, then you have these individual smooth muscle cells contracting independently. If you want sphincter control, don't use this kind. Iris, adjacent pupillary aperture, multi-unit, individual muscle cells contracting. So single unit, each little varicosity must release neurotransmitter onto individual cells, and they will contract if that neurotransmitter gets to the receptor on the smooth muscle cell. So each cell will generate its own action potential that will then open calcium channels, and the contraction process is initiated. So these swollen areas, they're named varicosities, like the dilated superficial blood vessels in lower extremities, you call them varicose veins. This is a dilated area of something, and this is a dilated area of a neuron process. It contains the vesicles of the norepinephrine neurotransmitter. Also has mitochondria there. We'll learn later in nervous system that the only way to break down the neurotransmitter norepinephrine, to take it back into the varicosity and break it down into mitochondria. So that's why you have mitochondria there, to take away the contraction, that action of the neurotransmitter. Since it's highly unlikely that this neurotransmitter that's diffusing in every direction is going to get back into a varicosity, it can diffuse to neighboring areas. If there's a receptor for the neurotransmitter, it can stimulate someplace in a far distant area in the body. So norepinephrine, it can have widespread effects, but this is a good thing because norepinephrine is in your sympathetic nervous system, which is fight or flight, and you want widespread recruitment. You don't want specific recruitment, you want widespread effects. So that's one way you get it, as well as recruiting different nerves. So single unit, this is going to be in visceral smooth muscle, so it could be in the heart, it could be in the GI tract, could be in the kidneys. This is responding more to the increase in fluid in the area, so it could be food in your digestive tract, stretch of the muscle cell will open calcium channels and activate contraction. Neurotransmitter can bind to a receptor and open channels as well. So you can activate it two ways. Most of the time it's the myogenic that is going to activate these single unit smooth muscle cells. So you put more urine in the ureter, then more squeezing to get it to the bladder. You put more urine in the bladder, then there's going to be a reflex to have the contraction of the bladder via nerves, and you void the urine. When you fill blood vessels with more cardiac output, then the smooth muscle in the wall of the vessel stretches and it contracts. That will keep the pressure applied to the blood, help distribute it out further. Each of these smooth muscle cells are interconnected by gap junctions. So you're going to see this term functional syncytium. Syn means to put together. To put together, to contract as a unit is a syncytium. When one area is stimulated by neurotransmitter, then via the gap junctions, the action potentials go to all the other cells, and pretty much as a unit at the same time, they will contract. Functional syncytium also in the heart. The heart contracts more or less as a unit to apply pressure to the blood to get it to move out into the pulmonary or systemic circulation. So the same functional syncytium in your urinary system and your GI system as well. So the cells are self-excitable, meaning they can be the cardiac muscle. Now we're in cardiac muscle. They're a functional syncytium as well. They have gap junctions. They stimulate the pacemaker cells. The heart has pacemaker cells like the smooth muscle does. Those send off action potentials into myocytes. Myocytes conduct action potential all over the heart. There are no nerves in the heart. There are nerves to the heart, but no nerves in the heart. All the myocytes have specialized jobs. Some of them act to conduct action potentials. Some act as pacemaker cells, and others act as contractile cells to create the pressure. The cardiac muscle cells in the SA and the AV node are self-excitable. They are the pacemaker cells. Their action potentials look very similar to the smooth muscle pacemaker activity. Slow depolarization, hit threshold, rapid depolarization, rapid repolarization. The gap junctions spread the action potential throughout the heart. We can skip that because it's just a review. So why so little about cardiac? Because we're not going to cover the heart till next semester. We'll cover a lot more about cardiac muscle cells and contraction next semester. Why do we spend time on smooth muscle? Because smooth muscle is going to be involved in the endocrine system and also part of the nervous system and spreading hormones and neurotransmitters around the body. Okay, so got 20 minutes. We're going to start on the endocrine anatomy, which I had open. Martini 19, yes.
You would say the most recent.
Okay. There's no number.
Yep, that's right, there's no number. Sorry about that. So anyways, you all know how to read an index. You should be able to find the chapter. Okay, so Martini 19 is endocrine system. So endocrine system's job is to save the nervous system a lot of work, a lot of ATP, save your body's metabolism from having to just continually shove food in, burn calories to make the energy to run your nervous system. And even if you were eating all the time, you probably couldn't run your nervous system, control all your metabolic functions 24/7. It would fatigue, there would be periods where you'd be blacked out and maybe You're out of touch with reality for a long period of time until you got the energy to then come back into awareness. So rather than fatigue your nervous system, you have the ability to have chemicals released, have widespread effects, or very specific effects. So cells that want to respond to the chemical, they'll have a receptor for that chemical. If they don't need to respond to that chemical, they won't have a receptor for it. But anywhere in your body that you have a receptor for that chemical, you get an activation of those cells. The endocrine system releases hormones. Most hormones are either peptides or proteins. Occasionally, they are smaller molecules referred to as biogenic amines, but most of them are going to be several amino acids in length and even as big as a protein, so over 100 amino acids in length. Although strict terminology says a hormone must go into the blood supply, there are also peptides and chemicals made of amino acids that just go to neighboring cells. Or the cell that releases it is then stimulated by it. So there are subclasses of the endocrine communication that are autocrine, which means the cell that releases is also stimulated by that secretion. So it's self-stimulating. Also you can have the autocoid. You can spell it either way, auto or autocoid. Those secretions go to neighboring cells to stimulate them. Some cells are both autocrine and autocoid. So they release something, they stimulate themselves, it diffuses to the neighbor and stimulates the neighboring cells as well. The strictest definition that I've seen for autocoid is that the secretion stimulates another type of cell that's in the vicinity. So a different tissue cell. But I've also seen it used as in combination with autocrine, where the cell is releasing the protein or peptide, it's stimulated by it, and it's stimulating other cells next door. Bottom line, if the cell doesn't have a receptor for it, it's not being stimulated by that chemical. Paracrine is where the chemicals stimulate similar cells, so like cells in that same tissue. Paracrines, autocrines, autocoid, they have very short half-lives. They very rarely get into the blood, and if they do, they're usually degraded before they get to any other tissue that has a receptor for them. So they're having localized effects. The endocrine hormones, they either have a transport protein which prolongs their half-life, or they have a long enough half-life that they'll reach their target organ and still be in a high enough concentration to have an effect on their target organ. So location, localized, this is widespread effect for the ones that go into the bloodstream. And then depending on their chemical nature, there are also subclasses of the endocrine chemicals. So eicosanoid, if they're made from arachidonic acid, then they're going to have a 20 carbon backbone. That's what eicosanoid means. If that arachidonic acid then interacts with cyclooxygenase enzyme, you get prostaglandins formed. So depending on the enzyme that's in the tissue, in the cell, you may get prostaglandins formed. But if there's a lipoxygenase enzyme, then leukotrienes will be formed. Thromboxanes are also part of prostaglandin production. So you need cyclooxygenase to get to thromboxanes. Each tissue, depending on the receptor it has, the same prostaglandin may have opposite effects in different tissues. It may cause constriction in one and dilation in another, or stimulate or inhibit. Depends on the receptor that it's binding to, what the effect is going to be. So any cell that doesn't have a receptor is not going to respond at all. But also you can have receptor subtypes, and that can vary the response to the same chemical. Amino acid derivatives means you're taking an amino acid and then sticking two together and making something like thyroid hormone, or you're modifying amino acids and then having another type of chemical as a communicator, or maybe it is just an amino acid that is acting as a hormone. Peptides means there are multiple amino acids in a chain. So 2 would be a dipeptide, 3 a tripeptide, you have a tetrapeptide. And then you usually get tired of saying that, so you call it an oligo, which means there's a few. Up to 99, you have peptides. 100 and over, you've got a protein. So the length of the amino acid sequence, if it's more than 1, and with the exception of thyroid hormone, which is 2, but it's not called a peptide, not called a dipeptide, it's called amino acid derivative. You have peptides. And steroids, those are made from cholesterol. So they have fat as their precursor. Steroid hormones, since they're made from fat, they are not water soluble. They get into the blood, they will be transported by a protein in the blood. That transport protein prolongs the life of a steroid hormone because it cannot be broken down until it's free of that transport protein. So on the transport protein, enzymes can't break down a steroid hormone. So if they get to the target organ, diffuse off and bind to the receptor or enter a cell, then they're good to go. Amino acid derivative or a peptide is water soluble, and so if it's in the plasma, enzymes can attack it. So they will have shorter half-lives. You gotta secrete more of them to have their desired effect. So amino acid derivatives, thyroid hormone. You start with tyrosine. And take 2 tyrosines and stick them together, and then stick iodines on them. You got 3, then you've got the metabolically active form of thyroid hormone. You have 4 iodines on it, that's fine. The tissues can chop off 1 iodine and have P3 metabolically active form of thyroid hormone. So 2 tyrosines stuck together, they're referred to, that hormone is referred to as an amino acid derived hormone. Even though it's really a dipeptide, don't talk about it that way. Take tyrosine again and have different enzymes act on it. You can make dopamine. You have another enzyme, you can take dopamine and convert it to norepinephrine. Have a third enzyme, you can take norepinephrine and make epinephrine out of it. So adrenal medulla has all 3 enzymes. It goes, you don't stimulate it very often, it will take tyrosine, convert it all the way to epinephrine. And when you stimulate your adrenal gland, most of what comes out is going to be epinephrine, some norepinephrine, and hardly ever dopamine. Yeah.
Is there an extra enzyme to make the L-dopa than the dopamine?
L-dopa is a precursor that's up in the brain, so different process, don't worry about it. Yeah, so you're gonna that L-DOPA is precursor to DOPA. L-DOPA can cross the blood-brain barrier, and then the enzyme can make dopamine out of it. So don't worry about that yet. Okay, so different process in the brain. So right now we're talking about endocrine, so we're talking about adrenal medulla. So it says norepinephrine and epinephrine because that's usually what comes out of adrenal medulla, which is 80 to 85% epinephrine, and then 15 to 20% norepinephrine. Very rarely dopamine. If for some reason you have a fight or flight response that is happening for a very long period of time, you're doing an ultra marathon or something like that, some of those freaky extreme sport things that go on and on and on, you have chronic stimulation of your adrenal medulla, you're going to see less epinephrine, more norepinephrine, and with chronic stimulation, you're going to start to see dopamine coming out. Why? Because you don't have time for the whole reaction to occur. So you've got dopamine made, but you haven't had time to convert it to norepinephrine. Dopamine will show up in the circulatory system. With repetitive stimulation, what norepinephrine you do make, there's no time to continue to produce high rates of epinephrine. So epinephrine concentrations go down, norepinephrine goes up, and you're starting to see dopamine in the circulation. Most of it's epinephrine because most of the time you're not stimulating the adrenal medulla. You got plenty of time to do that reaction all the way to epinephrine. In the nervous system, you don't see epinephrine because the neurons don't have that third enzyme. So they can convert dopamine to norepinephrine. They don't have the third enzyme to convert norepinephrine to epinephrine. So what the nervous system is releasing is norepinephrine. Some neurons don't have the second enzyme, so their neurotransmitter is dopamine. And you've got to get your L-DOPA to make your dopamine. Tryptophan, another amino acid, you make melatonin out of it. Melatonin controls your light-dark cycle, so your sleep cycle, also reproduction cycle. Peptides, got a chain of amino acids, probably longer than 15, less than 100. All pituitary hormones are peptides. So posterior, anterior pituitary hormones, all of them are peptide hormones. So it's the largest group of hormones. If ever asked, what is this a biogenic amine, is amino acid derivative, is it a protein or a peptide? You don't know, say peptide, you've got the highest chance of being right. Lipid derives all come from cholesterol. You must have cholesterol in your diet or your liver is going to make it for you. And if you have the wrong genetics and you don't eat your daily dose of cholesterol, your liver goes wackos and actually overproduces cholesterol. People who are vegans can die of atherosclerotic heart disease, and everybody's like, oh, how could that be? Because they ate no meat. But they had the wrong genetics, and because they ate no cholesterol, their liver went wackos. You give it a little bit, it's happy, it stays producing normal amounts. But you give it none, it goes crazy and overproduces cholesterol. So you can be a vegan and still die of plaque. And your family will be very surprised unless you know. And you have no way of knowing your genetics, really. I mean, you can try to get your genetic workup and figure that out, but more than likely, it's just better to eat a little bit of meat now and then, some cholesterol source, and you'll be fine. The adrenal cortex uses cholesterol to make glucocorticoids to control your blood sugar levels. It will make mineralocorticoids to control sodium and potassium. It will also make androgens that you can then convert into testosterone and estrogens. The hormonal stimuli can, so endocrine system, what triggers the release of hormone into the blood? It could be high or it could be low calcium. So the hormone, parathyroid hormone, responds to low calcium. Calcitonin is a hormone that responds to high calcium concentration. Insulin is a hormone that responds to high glucose concentrations, but glucagon is a hormone that responds to low glucose concentration. So there'll be different stimuli, and sometimes you have hormones that are antagonistic to each other. So one will respond to something when it's high, and another one will respond when it's low. Hormones also respond to other hormone stimulation. So hypothalamus will produce releasing hormones, which then release stimulating hormones from the anterior pituitary, and then they go out and stimulate the peripheral endocrine glands. So you have chain reaction, hormone releasing hormones, and hormone maybe recruiting another hormone and having its effect, or maybe then going out and having its direct effects. Enzymes break down the hormones and stop their effects, and therefore control the cellular response. And then cells have their own protective mechanisms by inserting receptors if they're understimulated, so they can bind more hormone, or if they're overstimulated, they'll pull receptors out of the membrane and control their overstimulation. So whatever their metabolic response is, if it's too high, they pull receptors out of the membrane. If it's too low, they'll insert receptors into the membrane. Hormones change the target cell's activity. They may turn it on, they may turn it off. It depends on what the hormone does intracellularly. So the hormone may get into the cytoplasm, it may get into the nucleus, it may be excluded from the cell and have to have a plasma membrane receptor. Plasma membrane receptors actually are more efficient at turning on or off intracellular responses, because that first signal, which is the hormone, is then amplified with second messenger signals within the cell. So one hormone could be multiplied in its effect in the cell to a millionfold. Steroid hormone that comes into the cell will then have effect on the genome, turning on the genome, transcribing mRNA, and then translating it, and then creating some response in the cell. Slow process. So the hormone gets in fast, but its activation of the cell is very slow. So where the receptor is will determine the speed of the response. If there's no receptor, the cell won't respond. If the cell is overstimulated, it may pull receptors out of the membrane. If it's understimulated, put more receptors up there, so it will get more binding of the hormone. Okay, so we'll pick up with